and biochemistry, DNA is a molecule of particular interest for a lot of reasons, and Dan's been talking about it for years. I've been researching about it for years, and since we are here at the same conference, he said, why don't you talk about DNA? So I put together 3 lectures from the past into one mega lecture. So either I talk fast or I reverse time. And one way or another, I'll get through it. Okay, so DNA exists in the body in many different forms, and most researchers work with circular DNA or linear DNA, and I'm actually working with the linear DNA and measuring the winding and unwinding of the 2 strands, amongst other properties of the DNA. But of particular interest, yes, for practical research scientists over here, we were having our discussion. I want to talk a little bit about theory because Dan's here, and the exotic properties of DNA are actually quite interesting and based in the scientific literature. So everything I'm going to talk about is from the scientific literature. For example, DNA exists in the toroidal form in the human DNA. I said that earlier. It kind of wraps itself around into this kind of a structure. This is an electron micrograph of actual DNA in a cell. And if they look close enough, you could see that it's kind of more like that. It winds itself circumferentially rather than that way around the toroid. And that it's a structure that's observed, it's reported in the biological literature, and biologists have no idea what the function of toroidal DNA is. Except I have an idea or a theory, and this is a model that I have presented 10 years ago, thank you, about the function of toroidal DNA. And it kind of would make sense to this crowd particularly because of Dan and your interest in Dan's research. So these are more electron micrographs of DNA in a toroidal function. Now, toroids, of course, you guys all know about toroids, but here's a crash course on the toroid. A toroid is another word in the physics literature for a toroid is a wormhole. And in the center of a wormhole is a catenoid. And this is the structure of a catenoid. And a catenoid has some very interesting properties, again, from the physics literature. And this particular article, they're talking about transitions between different states of the toroid. So it's not like it's a fixed shape; it's changing all the time. And one of the properties of this the center of the toroid, actually they call it the throat in the physics literature. The throat of the toroid is and another term they use it, transversible wormhole. Whoops, that's a worm, not a work. Sorry, typo. Transversible meaning that things can go in one end and out the other end. And what goes in and out is different forms of energy. And in the physics literature, they describe how the throat of the wormhole can actually allow one kind of energy to be converted into another kind of energy. Well, in my world, this is a possible mechanism for converting higher dimensional quantum energy into ordinary four-dimensional electromagnetic energy, which the body can use. So the DNA antenna hypothesis proposes that this is one mechanism for how higher dimensional energies, or spiritual energies for this group, can be converted into electromagnetic energy which the body can use. We all know electromagnetic energy controls biochemical and physiological processes, but this part of it is the missing part. So I developed this model which is based on geometric resonances, and the concept here is that this sequence of geometries it occurs in the body, so that, well, the twister is a toroid in a toroid in a toroid, and that's again in the physics literature. And then we can end up ultimately with a helix, and the idea is that the energetic shape of a molecule determines to what degree it will resonate with another molecule. And that whole concept is makes a lot of sense to you, I imagine, hopefully. But it's certainly not in, biologists don't think like that. So that's the conceptual model that I developed as one method of converting high-dimensional energies into another kind of energy. Now, that's one model. Model number two is phase conjugation. Everybody knows what phase conjugation is, except in the scientific literature, it's a little different than Dan describes it. This is a phase conjugation concept in the physics literature, whereby an ordinary conventional mirror, you have light, comes in, bounces off at an angle. In a phase conjugation mirror, the light that comes in bounces off and goes back to the source. And of course, in our language, the source could be God or some higher dimensional energy source. Now, the energy that bounces off is, in a normal mirror, is the same. E and E is the same. But here they have this concept of visualization where the energy that's bouncing back is different than the energy that's going in. And in the physics literature, the energy that bounces back is called virtual energy. And what this kind of a mirror therefore does is convert a laser or ordinary light energy into virtual energy. Well, gee, that's a mechanism converting one kind of energy to another. And then if you look even further in the physics literature, this process is reversible. So you can convert higher dimensional energy or virtual energy into ordinary electromagnetic energy, just like you can do with a toroid. So that's the physics. So then, being a biologist, I'm going, before I say that, also in the physics literature, particularly these two authors, everyone knows Edgar Mitchell, the astronaut, but Edgar Mitchell based his theory on Marcer, a Frenchman, who developed this idea of phase conjugation holography as a mechanism how an image outside the brain gets converted into the phase conjugate replica of the original image in the brain. And then, of course, the two images, the outer one and the inner one, are superimposed. So this is a quantum phenomena. And they talk about all this in the physics literature. So the idea of optical phase conjugation holography is a very interesting concept. And I'm taking this idea. I actually met Edgar Mitchell. I got involved with his second company after he set up Noetics Institute. He developed a company called Quantrek, and got all these scientists who were together, physicists actually, who were studying consciousness. And the idea was to extend this idea to other than just the act of perception, but other aspects of consciousness. And in honor of that team, I developed this theory, we're still talking about theory, sorry guys, about how optical phase conjugation can occur in the body. So the first thing I just wanted to say is that optical phase conjugation in biology is actually also used practically. It's used in photodynamic therapy. It's used to treat kidney stones with ultrasound. It's used in photoacoustic tomography imaging. So, I mean, this is, if you read in between the lines, these sophisticated biomedical techniques use phase conjugation technology. They don't talk about it much, but in the small print, it's there. So there is some rationale for looking at the role of phase conjugation in biology. So,
and Glen, Steve Lehar's somewhat famous phase conjugate model of perception.
Yes, well, okay, there's another one. I talked about Marcer and Mitchell, but there's a third person. So, yeah. Okay, so I'm not particularly interested in perception, but as a biologist, I'm asking the question, well, where is the phase conjugation mirror in the body? So if you look at scientific literature, people like Carl Pribram talk about neuronal circuits in the brain being the phase conjugation mirror. And then there's other people who talk about the spatial temporal patterns in the brain as being the source of the phase conjugation mirror. And of course we do know that crystals and liquid crystal lattices can function as phase conjugation mirrors. Okay, well, okay, so taking all that into mind, I searched the scientific literature to see whether there were any biomolecules. It's white on blue. I still can't see it.
Colorblind.
I'm colorblind. Yes, I am. Right. That explains everything. So the question was, is there any scientific evidence for phase conjugation in biomolecules? And it turns out that porphyrins, these exhibit this particular property. Porphyrins are, I'll explain them in a minute, but these are the two references for the experimentation demonstrating that porphyrin exhibits phase conjugation behavior. I mean, they put it in the test tube and they shine light on it and they measure the light that bounces off. And they talk about triplet excited states and they talk about nonlinear absorption. But the bottom line is this one chemical exhibits this particular property. Now, porphyrins, this is what they look like. They exist in certain heme proteins for any biologists in here. Heme is an entity attached to the protein, and the heme is made of porphyrins. They have different metals inside. This one is iron, but this whole system is a very complicated quantum system that allows for, because of the hydrogen bonds and the conjugation and the delocalized electrons, exhibits electron tunneling. So this is a quantum system which exhibits phase conjugation behavior. So that's in the scientific literature. So then I'm still asking the question, where is the phase conjugation mirror?
Well, that cross point across the iron is a good point. He means iron as in hemoglobin, right. And that cross at the iron looks very interesting.
Yeah, the geometry is awesome.
Four-way mixing right there.
Yeah, right, exactly. I love to see the three-dimensional structure of this thing, exactly. But I don't know that. I don't know if anybody knows that. Okay, so here's my one more theory before we get into the experimental stuff. Here's my theory that when you press the button, it goes crazy. It's not supposed to do that. Oh, there, good, okay. So, well, electronics has their own way. Okay, maybe this is what happens in the body, but what that there is a three-dimensional distribution of cells that are enriched with porphyrin. Now, it turns out that bone cells have a lot of porphyrin in them. So if you see each one of these dots when they're not moving as a cell, a bone cell, with a lot of porphyrin, well, that would create a three-dimensional pattern which could function as a three-dimensional mirror, as a phase conjugation mirror. So we're still asking the question, where is this phase conjugation mirror?
And does that look like negentropic behavior?
And is it negentropic? Well, yes, that's implied. I mean, that's kind of a given. So I don't even, that's a different question, but I'm assuming that based on your work. So here's a way of distributing these molecules in a three-dimensional plane, which could function as a phase conjugation mirror. So bearing that in mind, I'm proposing that exists at the base chakra, down here at the coccyx. Why? Because everybody knows about, somebody mentioned the Ida and Pingala. Well, in the center of those two channels is the forgot what it's called. This
Sushumna.
Sushumna, there we go, thank you, there we go. Which is a central channel. So at the skull, at the top of at the crown chakra, we have higher dimensional energies coming into the body. And it goes straight down to Sushumna, and at the base of the Sushumna is our phase conjugation mirror. And then, so here's the model is that as the energy, higher dimensional energy, universal light, whatever you want to call it, there's a beam splitter, and then this acts as a reference beam, and this acts as the
Interference beam.
The interference beam, and it goes through all of the bone cells that are surrounding the Sushumna. The Sushumna is in the center of the spine, which is therefore surrounded by bone cells. Bone cells have a lot of porphyrin. So as this beam bounces off the DNA in all the cells that surround the Sushumna, the two beams meet here and form the information patterns that are characterized, they characterize each individual. So this whole phenomenon occurs in the fetus. When the fetus first forms, the energy comes in, forms this pattern. And then, once you have the information pattern laid down, the same phenomenon occurs later in life, where the divine energy comes in, and then remember, it bounces back and it goes back up the Sushumna. And as it goes back up, the virtual energy is now coming back up the Sushumna. It spreads out and forms the biofield. So that's the model.
Glenn, just 30 seconds to play with that. When we measured Kundalini in the microwave, the ADP frequency, which is, by the way, phase conjugate applied, with Dratch from the spine liquids, we also knew the phonon geometry of what's superconducting that liquid from the work that Patrick's about to present in the physics of Kundalini. So we know the low-frequency phonons triggering that column, as well as the microwave frequency that's centered there, which is ADP, deoxyribonucleic triphosphate.
Well, but unfortunately, ADP or ATP does not function as a phase conjugation molecule.
Well, I approve that the wavelength of ADP is precisely golden ratio exponent times Planck, suggesting a phase conjugate molecule.
I take back my statement. No one's ever measured ADP, and if Dan's
well,
right,
1.91 angstroms in the literature on the primary dominant wavelength of ADP, actually, and that's what's in my equation.
okay, well, all right. So therefore, based on what Dan's proposing, ADP may also be a phase conjugation molecule.
That's under my chart in my book. Yes.
Right. No one's ever done that, but that would be a great experiment to verify your theory. And that would support my theory
exactly.
about how the biofield is created,
exactly.
made of virtual energy coming down by this mechanism. So
would that, sorry.
okay, so it's fast.
Would that be something that would be propagated by the environment? Because this brings me back to like some of Tesla's theories of like there can't be any matter without energy, in general. So like just the ether of space.
Actually, we're interrupting Glenn's talk, but again, 10
Please
seconds.
move on.
10 seconds. Bob Dratch took that microwave scanning technology, stepped out on the porch, and proved it was the ultimate dowsing tool. So we have proven that the conjugate wavelength in the microwave of the enzyme diphosphate, most important molecule in the body, is profoundly symbiotic with the Earth grid by measurement. So yes, it's a microwave bloodstream, among others.
Oh, that's great having you in the audience. I don't have to answer half of the question. Thank you, Dan. All right. So, for the record, this actually got published. So it's out. It's in the scientific literature. Okay. So, now we can go to experimental work. There are experimentalists over there. Okay. All right. Now, so, because I am an experimental researcher, so I'm looking at the effects of what I call subtle energy, or we call it phase conjugation, charge implosion energy, longitudinal waves, whatever you want to call it. The energy coming off the body. Now, the energy coming off the body is that, but it's also electromagnetic. So, as I said earlier, you can separate the two by putting the healer a mile away from the sample. But in these experiments, the first question was, can the energy from the body, as regulated by focused human intention, as Dan puts it, change the DNA at all? That's the first fundamental question. So here I'm working with DNA inside cells, and then we're going to work with just pure DNA. So this is DNA inside of either tumor cells or lymphocytes, and in a petri dish. That little thing there in the sphere is a petri dish. I've done a lot of research in this, and with the interest of time, I will just summarize the essence of the research. So we're measuring DNA synthesis, in essence, which is how ends up causing the tumor cells to grow or not grow. And I work with different healers with different intentions. And there's always a control, and then, do this scientifically, measuring thymidine incorporation into DNA, radioactive thymidine incorporation into DNA. It's a standard biological measurement for growth of cells or DNA synthesis. The radioactive thymidine gets incorporated into the DNA, and then you can measure how much the DNA turns over, which is a measure of its ability to replicate and synthesize itself. So that's a bioassay. And it's worth mentioning. So when I say increase and decrease synthesis, that's how it was measured. The healers then go through different intentions. And depending on the intention, this is with tumor cells. They can either increase or decrease the synthesis, DNA synthesis. Some intentions are more effective than others. Focused thought, which some people would call mental healing, doesn't really work. You need the heart component, or you need the coherence, I guess. You need the love, the next line. And the heart without the intention doesn't work. You need them both. It's not just one or the other. And then I had another healer who could do imagery work as separate from intention. And if just the imagery is about as effective as the imagery, but when you combine the two of them together, it's even more effective.
Glenn, this is so fabulous, just again, 10 seconds. So in a few hours, Patrick is going to present the difference between front brain and back brain EEG coherence,
Okay.
and saying that in the flow is always back brain. So what you're saying is the front brain alone won't do it, but we'd love you to see when the back brain's in the flow.
Yeah, there's a lot of research to do.
This was linked as well. So
This was done 10 years ago, and way before we even knew about this kind of esoteric stuff.
This is late in mind.
That was done with a healer called Leonard Laskow, who wrote a book called Healing with Love. And
how were you guys measuring that?
measuring the incorporation of radioactive thymidine into the DNA molecule, which is the standard biochemical measure. Are there any biochemists in the audience? Right? Probably not. How you measure. Fair enough. Okay. So that's one set of experiments. Here's another experiment with the same guy. So instead of directly healing the tumor cells and measuring the growth, I had him treat the tissue culture medium. Well, actually the water. And then I make tissue culture medium. You add proteins and amino acids and fatty acids to feed the cells. And then you add the water to the medium to the cells, and you get the same effect, but it's not quite as strong. Imagine that. The water stores the information, but not 100% of the information. But nonetheless, it did work pretty much, similar, literally. It was a 40% inhibition versus 28% inhibition.
Oh, yeah, just with intent.
Indirectly via the water. Okay, so that was a fun experiment.
Focus your intent.
The intent was, in that case, the intent was to slow down the growth of the cells, which was what happened. Okay, now we're moving to another whole series of experiments with purified human DNA. That is a healer. Okay, now, in this case, I'm measuring the winding and unwinding of the two strands. So, DNA normally looks like that, on the left, upper left, but it can unwind, and it can wind even further and make a kind of a tight knot. Both of these processes occur in the body. The top one occurs right before a protein or an enzyme binds to the DNA molecule. It kind of opens up and says, welcome. And then the molecule binds. So you can't really say one's good and one's bad, which is why I stopped doing this kind of research, because everyone kept saying, well, is it good for the body or not? I don't know. They both occur in the body naturally. And what holds the two strands together are hydrogen bonds, depicted on the right,
Very attractive.
between the two bases.
The black hole at the middle.
The black hole in the middle. Hydrogen bonds are actually quite interesting because they exhibit quantum behavior. Quantum chemistry has observed that the hydrogen bond, because of the delocalized electron and the cooperative interactions, exhibits quantum tunneling and quantum entanglement. So by measuring the added presence, when it winds together tighter, there are more hydrogen bonds. So you're introducing more quantum properties into the DNA molecule because there are more hydrogen bonds.
And remember, we choose to call or think about perfected entanglement, perfected embedding as named conjugation of stasis.
What he calls embedding, I think, or braiding is the same thing as winding.
But actually, if you think about entanglement, a little donut never gets inside a big donut successfully unless you use golden ratio conjugation. So that's entanglement perfected by definition.
Okay, that's entanglement between two molecules.
Yeah, so entanglement perfected is embedding perfected, which is actually conjugation perfected.
Which is what I'm
Fits the
talking
whole line,
about
yes.
in a different language.
That's right, slightly different.
Okay, so these experiments were done at Institute of Heart Math, whereby I was doing this work and they got all excited about it and they wooed me, and I left Harvard, Stanford University to go join Heart Math. The best move of my life. I left academia and bye-bye. And that was the end of my research with academia. So here's an absorption spectrophotometer. You measure the absorption of light at a particular wavelength, 260 nanometers, and there's a big peak because there's a chemical inside the DNA that absorbs the light. And the more the DNA is opened, the more light is absorbed, and these peaks go up. And if it closes down, it can't absorb the thing inside that absorbs it, that absorbs less. So that's the concept. So you have DNA in a test tube, you give it to someone at Heart Math and say, here, go into heart coherence.
Go love it.
Go love it. Send some love to the DNA and see what happens. So, and this is what happened, and then they intend to unwind or wind the DNA. So here, this large increase in the peak indicates unwinding, and here a decrease indicates winding, and here this is an experiment done about a mile away when the doc, who's the founder of Heart Math, Luke Childre, thank you, did these experiments, and he was about a mile away, and I'm in the lab, and we're talking on a wireless, okay, doc, I'm ready, send the energy, okay, and then I took a measurement.
Can you explain the function of the control in these specific graphs?
Well, the control actually was done beforehand, and then I call doc and say, okay, I did the control, now I'm ready for the treatment.
Right.
But that's a good question.
So the control was when they weren't sending
When they were not sending energy,
Okay.
and I said I got to set it up first.
Right.
The experiment. Then I did the controls. Then I called them on the wireless and said, okay, doc, I'm ready. Okay, but good question. Okay, now the point here is that since they were doing these kind of measurements with the HRV coherence, when there was a successful transfer of information and a change in the structure of the DNA, the EKG patterns were doing this, and when they were doing the top, there was no change in the DNA. So this is where Dan's famous statement comes from.
Patrick, is that wavelength on the bottom the Mayer wave, Patrick? Is that HRV? And the duration there, one has to be about Mayer wave intervals, right? Because they teach 1 hertz. So they're doing the Mayer wave and calling it radiation.
I never did we could do that maybe with the software these days. What is the frequency of
Well,
that.
it's indicated, you got 50 seconds and you got 5 peaks, that's 10 seconds per peak.
Oh,
We're
there-
done. We
oh,
just
okay.
spectrometer.
Well,
It's
thank you.
0.1 hertz. I just did it. I finished now, it's 0.1 hertz.
There we go, right. So this demonstrates that in order to see this effect, you need coherence
And this specific is meter wave coherence.
in the heart. Right,
Which
okay.
is what they call coherence, this is one frequency only.
So that we're going to call entrainment. And then the question is, if you've got this heart coherence, what happens when you focus your intention or not? So with no intention, if you look at the second column, 4.1 and 4.3, there's really no change in the DNA. But if there's intention and coherence, then there's a large
Nice.
influence
That's nice.
on the DNA.
That's good.
So, important observation.
Wait, what's N? Sorry.
N is the number of experiments.
Oh, got it.
And then SD is the standard deviation, and P is the statistical significance. So if you do it enough times, you can get statistical significance. Okay. Okay, so got through that. Now, this is all experiments where you take a measurement before and a measurement after. Not kinetics, but just a single time point. And of course, DNA changes a lot, so you get a lot of variability. So then I decided, well, okay, let's do some kinetic measurements where I'm taking a whole series of measurements. So how do you do? So in this kind of experiment, we're measuring the self-assembly process, which is itself a quantum property. Because the DNA, if you separate the two strands, they come back together automatically. That's the intelligence of DNA. But the way I did it was not to separate the two strands completely, but to separate them partially. So in this, and you use heat to do this. So then this particular system, it can go either way. It can keep unwinding, or it can rewind back to the original intact helical structure. And you can measure that as the absorption of light as a function of time. Because as it rewinds, there's less absorption because the thing that's absorbing the light is protected and there's less absorption. So what you get is a slope of this curve is what we're measuring now, which is a measure of the rate of rewinding. Okay, Dan gets it, I'm assuming everyone else
And
gets
we're
it.
saying that
I'm
the
looking-
winding process is centripetal in opposed to the unwinding
Here
is
we go.
opposite.
Okay.
And centripetal is clearly what happens during love or bliss. Your DNA gets thicker, actually.
So if the photons are being absorbed at this point, can you measure the photons being released?
Released.
Do they get released? When
Well, that according to Pop, in fact, what Pop did was he, instead of using heat to do this, he used a chemical called ethylene bromide, which causes also the same phenomenon. It causes the two strands to unwind, and what does happen, that Paul's pointing out, is that biophotons are released. So I'm not actually measuring the release of the biophotons, I'm measuring the actual rewinding of the two strands using the slope of this curve. So, here's the kind of results with several different kinds of healers. The control is the bottom line with an average slope of 0.42 plus or minus 0.2. There's some nice numbers for you experimentalists. Well, Reiki practitioners slow down. This is, when it goes that way, it's slowing down the rewinding. And they're all different, but Reiki practitioners were actually very good at this, and they slowed it down to the point where it's hardly rewinding at all. So then you would ask the question, well, why would a healer slow down this process? I would expect a healer to speed it up. This is a natural process of putting the DNA back to its intact form. You would think a healer would speed it up. Well, these guys all slow it down. And why is that? I mean, we could talk about that, but I think that what's happening is the fidelity of the process, the accuracy of the process is increased by slowing it down to make sure that the right bases combine and it reforms correctly.
To the proper speed.
So that's my explanation.
Yeah, because earlier our physics was suggesting it was related directly to the phonon geometry. The phonon, the sound standing wave, which is bliss basically, which is integral to coherence of the EKG, is a primary mechanism in the braid-on-braid process. So that would be interesting to measure.
Well, that would be interesting to measure,
Yeah.
but can that explain this phenomenon?
The phonon coherence is a sacrocranial still point, the healing
They
crisis.
call it the still point. Because
Still
for
point,
all
okay.
ways-
Okay, thank you.
That's
What
That
happened
explains-
when-
That's,
Yeah,
yeah, exactly, yeah, yeah, thank
when
you.
you
That
go
is,
inside
Oh, no.
therapy,
And the
it's
third,
same thing.
yeah.
well, we
The
could try.
moment that you put the power on,
Right.
and then a few minutes after it's coming back. But it can take seconds and it can take minutes.
And the idea is, if they make the still point, they emerge in greater phase order.
So if there is no wave anymore there, nothing could happen because waves are moving everything.
And that
So
slows
it
down.
can be an
Yeah.
explanation that it's waving.
Yes, yes, thank you, Patrick. Excellent, very good. And in fact, that would be a great experiment to see what the Therify does.
Well, we know that therefore the majority of people have a sacrocranial still point there.
Right, so that should slow it down.
They measure.
Let me see what's next before I go prepare myself.
Before we interrupt you too
Okay,
much.
no, all right. So then I was interested in some, everyone remembers the Vibe. I said, well, that's what healers do. Healers over here, and the best healers are on the far right, and the good healers over here. And then I was, what would technology do to this system? So everyone remember the Vibe machine? It's an inert gas technology where not two tubes, but they have a whole bunch of different inert gases, plasma tubes in a circle, and the whole thing radiates a thing. Well, that also slows down, not as good as healers. Interesting. And then, and this Ultera powder is a paramagnetic mineral. You just put the DNA, in a little, the bottom of the test tube in a little pile of powder, and you do the experiment, and it radiates paramagnetic energy. Works, but not as good as a healer, so I was very happy with these results. Talk about, my consciousness affecting the results. I was praying that it would come out this way, right? I'd like to think that healers work better than machines, and thank God, it worked out that way.
So Therify should perform better than Vibe because of.
Oh yeah, Therify would be,
Yeah.
Therify maybe.
I'm hoping it's over
And
there.
then far right, yeah.
I'm going to influence next.
And you can disprove, make me miserable again. Like, oh God, the machine can do just as well as a human. Right, but anyway, okay. So then, in the meantime, I met the folks from the Reconnection.
Oh yeah.
The piece of California. You guys probably know about the Reconnection. It's a new group of healers that claim that they're bringing in new energy onto the planet. And I said, really? Well, that's cool. Let's see what you do. So I did this study now with 4 different practitioners, including Eric Pearl himself. I just brought they were in New York. I was living in New York at the time. Brought all my equipment to New York, to the hotel, set everything up, and at 4 practitioners, we did experiment. I did the controls first, and then I bring in the practitioners, and guess what they did? There in the bottom, they speeded up the winding of the DNA. The curve in the very bottom is in the other direction.
What did they say about that?
They said, well, of course, we're bringing in new energies. So they were...
So they're not like, okay, yeah, distinct difference between slowing it down for the healing process and actually like importing, I'll use the word, importing new energy. So
which apparently has a different effect than most of the other healers. Now, I haven't done all the healers on the types of healers on the planet. I mean, Patrick, or God knows what Patrick would produce, or Sandra, is also a healer. Probably a lot of healers in the audience. But so far, most healers slow it down, and the Reconnection folks speed it up.
Reconnection is all over Italy. It's big.
Oh yeah, it is? Okay, yeah.
Are the Reconnection folk also kind of promoting healing first before getting all the new software?
They're
Or instead of overloading?
I don't know.
I'm just curious. I haven't talked to them.
But what this implies to me is that there are 2 kinds of healers, healing energy. One that stimulates the system and one that slows it down. So therefore the question arises, as a person who needs to go to a healer, what kind of healer do you need to go to? Do you need to slow your system down or speed it up?
completely individually based.
Or do you need to do one first and then the other? Or, so these are all the questions that arise
Very valid question.
from doing this kind of experiment. Yeah, right. Well, And we need funding, so there we go. Okay, to do this, because right now I'm doing other things. Okay, so now, since I am on the board of the U.S. Psychotronics Association, I had to do some kind of psychotronics research, radionics research. So I used this, I did this in collaboration with Peter Moscow, who okay, it's a little complicated, but he's 1,000 miles away with this. I sent him a sample of the DNA. So I've got a sample in my lab in California. He's in St. Louis, which is 1,000 miles away. And we start the experiment at the same time. He's measuring the etheric properties of DNA with this thing. And I'm measuring the winding and unwinding with a spectrophotometer. And we had to stimulate how did we do? We stimulated, that was right. I stimulated in my lab the DNA with the Vibe machine. So it's a plasma energy exposed to DNA. And then you see the two lines. That's Peter's measurement on the top and my measurement on the bottom. Remember, the two DNAs are entangled. They're 1,000 miles away, but they're entangled because they come from the same source. So what's interesting is that the peak at around 9 minutes after the experiment right here was the same. They both got the same response. But what's even more bizarre and interesting is that he saw a peak here with the etheric DNA, which was not present with the physical DNA. Yet. The physical DNA didn't respond until down here. The etheric DNA responded both here and here. The stimulus was the plasma wave from the device. It was 1,000 miles apart. I'm in California and he's in St. Louis.
Did you go further in time? Because maybe further in
No,
time you get that.
there's a lot more I could do. And this is what we did. These are the results we got. Sharing them.
And you'll do the next study as soon as you get the check.
Yeah, exactly. Okay, so there we go. Okay, so that was the fun experiment with radionics. Now let's talk about the electrical properties of DNA. Now, Dan already mentioned the superconductivity. Well, the superconductivity happens when electrons flow down the central channel of the DNA. And this has been measured. They measure the speed about how fast it takes. Now, both electrons and protons go down, or they go on the outside as well. They propagate on the outside, and normally they jump from one charge point to another. They hop. It's called electron hopping. But they can also tunnel. So electron tunneling is the quantum property that also occurs in DNA. It's in the scientific literature. It's all published. Okay, so bearing that in mind, what I did in these experiments, Cleve Baxter. Everyone remembers Cleve Baxter. Well, his apparatus that he stuck in the yogurt, electrodes that he stuck in the yogurt, we did this at HeartMath, and we stuck the electrodes in two different solutions.
This is Secret Life of Plants.
Right, that guy. Right, but his famous experiment is with plants, and the electrodes on the leaves, and then you harm one plant, and the guy who's wired up freaks out and responds. And you can't harm my buddy, and freaks out. So what we did was same electrodes. We got two different solutions. One was just DNA, and one was tumor cells. So what we did, the setup was we had them running on two different computers, and you could see in real time, the electrical activity, and then we're having a conversation. And we're monitoring the screens. How is the DNA responding to this conversation, and how is the tumor cells responding? And it's quite interesting. These are the kind of results. Oh, here's the setup that you get. A standard amplifier, ADD computer, blah, 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 standard stuff. Okay, so the kind of responses you get are you can get this kind of response, where the voltage drops, or you get this kind of response where it goes up. Okay. Okay, so then when you monitor them both simultaneously, you can see the control, it's pretty flat. Nothing happens most of the time. And then in the case of the tumor cells, we're talking about it's spontaneous. You can't really plan this. You can't even make it up, as the expression goes. So we're having a conversation, and all of a sudden we're talking about somebody who's got cancer and just got diagnosed, one of our colleagues. The tumor cells completely freak out and get that response. And the DNA is not responding. And then in another conversation, we were talking about genetic engineering or something. It just came up spontaneously, and the DNA freaks out, and the cells do not.
Somebody's listening. But it's not big brother, it's little brother.
Right. So these were fun experiments, but you know they say a lot about consciousness and intention and all that. So those were fun experiments.
Little brother.
Little brother. Okay. So the last series of experiments to talk about is the resonance frequency spectroscopy, which I spoke about in the first day. This is taking this slightly different setup. And I guess most of you heard it the first day, but basically you're measuring the electrical conductivity of DNA. You apply a voltage measure, the response at specific frequencies. And here's a typical graph that you get from this kind of a measurement. And you can see there are certain peaks at specific frequencies. And particularly like here's a nice strong one at 6 kilohertz. Okay. So then the question is, what happens when you, in this kind of a measurement, you expose the DNA to different subtle energy technologies? Okay, well, here's a spectra of the frequencies of DNA. So if anybody wants to know what the frequencies of human DNA are, here they are. And there are many of them, and some are stronger than others, obviously. Okay.
It's kilohertz there.
Kilohertz, they're all in kilohertz, right?
Your 6K one's not there then.
Yeah, well, not there. I started it at 10.
Yeah. But the 6K was the highest peak.
Yeah, well, no, there are higher ones when you go even higher.
Yeah.
So there we go.
Check my chart.
Okay.
I'm going to check my equation.
So then here's an example of what happens when you put a cell phone or DNA on top of a cell phone. And you can see in some cases there's a large drop at the lower frequency, and then here a really large drop at 75 kilohertz, and another large drop, and another large. So at higher frequencies, it's particularly detrimental. But at some frequencies, there is no effect. Okay. Now this is typical. The sensitivity of DNA to subtle energy is frequency dependent. It depends on the frequency that we're measuring.
What you'd expect.
Which you'd expect. Right. Okay, so here is Sandra's system, where here you can see at certain frequencies, like 39, there's a huge increase, and another large increase, and some frequencies there's no change. Back here too. So this is another way of measuring the effects of scalar waves, in this case from Sandra's system, on the DNA molecule. Okay, here now we're going from a machine to a healer. The data is expressed a little bit differently, but you can see that these are two different healers. So at this particular frequency, they both get a peak, but this healer is better than this healer. At higher frequencies, this healer produces an effect at about 60 kilohertz, and at about 80 kilohertz, the other healer produces an effect. So different healers have different effects on these frequencies of DNA, and you could almost use this as a marker to say what kind of effects each healer has. Each healer will have a unique response.
And if we had the key signature of the EKG or HRV, we could compare it.
Correlate, right. Okay. There's a lot of further research to do here. Okay, we're back to a machine again. This is a machine called the Trinfinity 8, which is designed to help people spiritually. It's a complex thing. Here's the actual experimental setup.
Came to the first
this is her device over here set up with this kind of setup, and then out of the machine comes these two quartz wands that you're supposed to hold. Well, DNA doesn't have hands, so I figured, well, I'm going to point them at the DNA and see what in the middle. So that's the experimental setup, and here's the result. Now she has different programs. So she's got a program here. The control is about there, about 15%. The aging program that she has caused a larger effect, but the program called the divine alignment did not affect the DNA. So error bars indicate the experimental variation. So that was interesting, but nonetheless, it's a subtle energy technology, and exactly how she does it is proprietary, but it's a combination of crystals and phase conjugation, scalar energy, and God knows what. Okay, here's another technology which is based on spinning geometry. Their website is I Am Evo, and it's a technology that they use to raise consciousness. And what they actually do is they take this spinning geometric structure and they put it. You're lying there like this, a bit like the Therify, and above the crown chakra, they've got this spinning thing. So I was saying, well, a spinning thing radiates some kind of energy. What does it do to DNA? Oh, you can see it involves geometry, spin, color, crystals, scalars, Fibonacci, tone, everything you can think of. And these are the clinical results that they observe from people who lie in the machine. And they've got these questionnaires. They measured, their the ability to focus and mental clarity and emotional balance and stuff like that. And these all increase according to their measurements. So this is a great technology to say, okay, give me one of those things. I'm going to bring it back to the lab and measure the DNA. So these are the results. At one particular frequency, there's this huge effect to increase the electrical properties of DNA. So that technology resonates with DNA at that particular frequency. Voila. Other frequencies, no effect.
1.41 says 1.41.
This is what I observed. Each technology resonates with DNA or not at specific frequencies.
If you could spectrum analysis of their emission.
Yeah, we could do all that, absolutely. Right, so again, to see what the Therify would do in a similar situation. The more potent energies affect DNA at many frequencies, not just one. But this has a particularly large effect. So the goal, large effect at many frequencies.
1450 is precisely phase conjugate to quantum.
All right. Okay. Okay.
That's
So,
pretty close.
there we go.
We've got time for questions.
So we got more questions.
Plus, don't leave the room while the questions are on, because after the questions I want a favour, and that is everybody over here for background group photo. So we'll do the questions first, and then because some people are leaving after lunch, so this is our only opportunity to have a photo. So questions?
So Glenn, it seems like from a lot of your results that the DNA is just like this wide bandwidth antenna. Seems like a transceiver too, because if it's receiving the energies and you're measuring the differences, it must be resonating and transmitting.
Well, it certainly emits biophotons, and if the DNA, and all these experiments were done with linear DNA.
Okay.
So imagine you can actually generate toroidal DNA in a test tube. Experimentally, there's a way to do that. So the next series of experiments I would love to do would be with toroidal DNA, because then, yes, it will really be acting as an antenna, as a phase conjugation system. All the results should be enhanced.
So Paul's the other thing, it's both transmitter and receiver. That's the point. It would be better if it was toroid.
Yeah, right.
Cool.
I don't know if this is going to make sense, but how are you able to work with living DNA?
Well, you buy DNA from the local drugstore. Actually, it's a chemical supply store. The DNA comes from human placenta. It's not, it's living or it's not living, it's not really living. I mean, it's purified from human placenta. And they get rid of all the fats and the lipids and the amino acids and the proteins and the garbage. You just got pure DNA, which you buy. And you take it up in water. So you've got DNA floating around in water. Now, this is a very artificial setup. It's not like the DNA in the body, in a sense. It's just DNA floating around in the test tube.
Right.
Is it living?
That makes
I don't know.
Well, I mean, yeah, it must be conscious.
Well, yeah, if we all believe that consciousness is in everything, there is consciousness even in a molecule. Even in that crude, non-living state, it exhibits quantum properties.
Because my question is,
It could be entangled.
would it be possible to do these experiments on a sample of DNA from a dead carcass, for instance,
Or better yet,
or a bone from a prehistoric being?
Well, see, again, if it's not pure DNA and you've got proteins and everything else in there, then you don't know what's causing it, what's changing, and then if it's got a protein right next to the DNA, the protein absorbs the energy and the DNA doesn't, or it makes it much more complicated.
Yeah, that's a
Right.
complex.
And I mean, ultimately, I like to do this work with people,
Yes.
and then take their DNA and see how their DNA changes. But that's a lot of work, and then you have to purify the DNA because you want to take a measurement, and it's the same problem. You're taking a measurement, and there's DNA and protein and everything else in there. What are you measuring? And they influence each other. So you'd have to then take the blood from someone, purify the DNA, and then measure it. So it's complicated and expensive. So I haven't worked with people. These experiments were just designed to see if DNA as a molecule would resonate with these kind of energies
Well,
at
yeah,
all.
that clarifies it in a general sense.
Yeah,
It'd
yeah.
be interesting to take that experiment.
Absolutely, to the next level. Absolutely.
Terry, you have a question?
Yeah, this may be show my lack of biochemistry, so I apologize, but couldn't you do what do you think, if you did the same type of thing with the so-called junk DNA, it theoretically would respond the same way?
Yeah, good question. No idea. Well, if we believe junk DNA does more than make proteins, then yeah, it should be in principle, because we all believe that the esoteric properties of DNA are in the junk DNA, so it should respond better. Yes, it should, in principle, yes. Now,
Yeah, these effects are all related to frequency, therefore wavelength. I was wondering, going back to the Vibe machine experiment, did you keep track or decide which of those spectrum tubes was facing the DNA? Because the Vibe machine is a foot-long vertical tubes.
actually, what the geometry in the Vibe is these things all around the circle, and I put the DNA on the top. Because I didn't know, and they're all different inert gases, so I didn't know
On top.
which gas was going to emit the right energy.
This then is on top of basically a Tesla coil.
Yeah.
That'd be a
It's
lot
like these tubes are actually acting as a collector
of potential.
of energy.
So that's a test for a longitudinal.
Yeah.
Oh, there's, when you talk about your longitudinal coils, because that's what you built.
Yeah, yeah. But that's a great question, because the energy coming off of a system like that is not homogeneous.
No.
We just saw the nodes that Dan just showed us. So you got to get the right distance too. If you're this far away or that far away, they can make a difference.
Well, that's where the peak energy of the Tesla coil is, on top of the end of the line. So that's very interesting.
Yeah,
Conjugate is direction.
yeah, right. So I typically, to do an experiment like that, I would typically use my pendulum and hold the damn DNA and say, is this the right position? This is I got to put it here.
That's good physics.
And for this group, I could share that. For most scientists, I can't.
Patrick had a little fun.
Did you try with a piece of music?
Music,
Yes.
Oh, yeah.
music, sound, music.
Changing
I've done so many
Many
experiments.
Yeah, bird sounds.
Sound.
The nutrient uptake in bird sounds sounds good.
coming from nature.
Yeah. I'm trying to remember. I mean, it's like 10 years worth of research I must have done sound at some point. Oh, yeah, I did. Suzanne Alexander made a tape called something, based on the infrared frequencies of DNA. She took the frequencies from the infrared spectroscopy, converted them down into the audio frequency, and used those notes to make music. DNA sequencer, I think it's called. It's a CD, it's an LP. And yeah, I don't even know what date it is, but that also affects the
Well,
DNA.
there's the famous Ono music from Japan, who did DNA music, one of which sounded like Chopin, actually.
Really?
Yeah, the Ono music, ONO DNA music, Japan. But that's a,
Oh, okay. Well, that would, yeah. Again, there are so many different kinds of stimulus that you could use, and obviously for this group, we're interested in the energy from the therapy. But yes, so sound energy. I think it's more the question of the frequencies. You know.
it's cool if that was 1450 was on the.
Well, yeah, really. Thank you.
Any more questions?
My question, the question of frequency, because in a piece of music,
You got everything. Yeah.
the frequencies are moving.
Yeah.
So there is a spin.
Yeah. Right.
You understand?
Right. Yeah.
And according to the graph, you say sometimes with some device on singular frequency,
Right.
but with a piece of music normally,
Yeah.
is that
Yeah.
it
Yes.
moves
Yeah, yeah,
inside
yeah.
with
Yeah.
the music of the
Yeah.
orchestra.
Yeah.
Well, could it be?
Thank you